So our next speaker is Arvind Ramanathan from Argonne National Laboratory.
All right. Hope you can hear me. Yeah, I guess. That's good. Thank you so much for the invitation. And you'll be hearing a lot from our group talking of foundation models and what we are trying to do there. But I wanted to focus on biology because this is something that we do in the lab. And this is something that we are very excited about as to how AI is actually changing the scope of what we can understand about biological systems. And this is by no means one person's work. It's a lot of different people's contributions. I'll acknowledge them at the end of the talk. But the motivation actually started by thinking of cells and systems as, constituting different parts that are being put together, right? If you think of it like a car, you have these car parts that you want to describe and define and design them. And so a cell is usually made up of all these different parts, including transcription factors, enzymes, and enzymes that are together to do certain things nicely, genetic parts as well. And then as part of this project that started out with DOE for designing what we call secure biosystems, meaning that you want to design these systems in a way that don't leak out into the environment and do bad things, right? How can you build these things in a robust fashion? We needed to do this in a rather rapid way inside our lab, right? And as part of this project, much of this actually impinged on the fact that we could design these things using computer suggestions. And we wanted to be focused on the aspect of reproducing everything that we did in the lab. And that's actually one of the main motivators for saying, is there a way for us to actually understand what we think of biological systems? Meaning, can we build effective representations for biology? And one of the fundamental challenges about biology is unlike other subject areas like physics and chemistry, where there are certain rules that we sort of well understand and sort of apply mathematical principles to design these things, biological systems have a very different sort of a feel and form to it, right? They agree and they actually base on certain physical rules that we know, because laws of chemistry cannot be violated and so on. But we wanted to see if we can actually understand the code of life using these sort of models. So most of foundation models, at least in my belief system, is that they are built towards building effective representations. Can we learn effective representations from a large amount of data? And this has to respect the intrinsic hierarchy of what biological systems are, and also leverage all of the rich amounts of data that we have access to today. And then we come down to this factor where we can build out generative models so that we can design them using experimental feedback, which is actually one of the fundamental ways in which we can understand these systems, and then eventually also to generate synthetic genomes. And I'm going to show that in the context of SARS-CoV-2, where we were able to predict future strains without having to look at a lot of the data. And then finally, how do you use these inside the lab to guide your experiments, which is the place where we actually want to say how we can embody these agents with certain tools and actions that they can take. And this is towards designing a self-driving lab for discovering antimicrobials. And I'll also show how the AI can sort of generate code on its own to write protocols inside the lab and automate them in that context. So the first portion where I'm going to talk a lot about this genome scale models. This actually started out with this very nice pipeline that Karpathy had put together about saying, what does it take to actually pre-train on all of the data that we have? I'm going to lay this out because this is going to be, again, reflective of how we are doing our tutorials. Also later on in the day, you'll hear a lot from my students as to how they have been thinking about it from the context of pre-training to supervised fine-tuning to reward modeling and reinforcement learning, which is the last portion of my talk too. But this is going to start with the raw genomic data that we got from this database called as PATRIC. And if you have not heard of PATRIC, it is one of the largest resources for bacterial and viral genomes. We are mostly focused on prokaryotes because I think prokaryotes have a lot of things to teach us. And so we are using prokaryotic genomes as the starting point. And then we'll do language modeling on them with these base models that I'm going to talk about. And then we'll talk a lot about the applications and the results. So The hierarchical information that is contained within the genomic data is kind of cool because you actually have like various factors that you need to take into account, from starting with the simple idea of what a gene is all about, which has the open reading frame, the 5' UTR and the 3' UTR, start and the stop codon. You have all of this information encoded in this piece of DNA, but a single gene can then organize itself into viral genomes or plasmids and pathways and bacteria and eukaryotes all the way to humans. All of this information still carries a very small vocabulary, which is just 4 letters if you just take the ACGT for DNA-based characterization. And if you take RNA, you have ACGU. But this is still a very limited vocabulary when you start at the level of the individual nucleotides. And when you start putting together nucleotides together to form words, which are usually like codons and so on, you start seeing more interesting stuff happen where the vocabulary starts to increase quite nicely. And then you have open reading frames, which are now collections of these genes that are put together in different orders. And entire genomes are just nothing but collections of all these, full set of genes and the open reading frames and everything else together. So we needed a model that sort of allows us to capture this in an iterative fashion. And this is actually the genesis of the first model that we started building, which was genome scale language models. And again, I call this as first because this is not done and hashed out. We are still learning a lot from these models as to what their limitations are and how we are thinking about them. So the input sequence is usually broken down as codons. You can have various representations for the codons. You can have a continuous representation, which means that you're actually doing a moving window. Or you can also do this as discrete codons in the way that I've represented it here. You're taking these codons and putting them through these transformer layers, and the transformer layers basically learn what is called as a semantic embedding. This is all basic transformer architecture. There is nothing special about these architectures per se in terms of what we have done. And at the end of the day, you can actually emit these probabilities that basically tell you what is the likelihood of seeing a particular position with that codon and so on. Then you can translate that back into any protein of interest and so on. The idea to build this model using these semantic embeddings resulted in this foundation model, which starts out with this base model with a sequence length of 2048 codons, right? And we can train it at the level of 25 million parameters all the way to 25 billion parameters. And this data set is still the same. We use the Patrick BVBRC data set, which is 110 million genes from all of prokaryotes that we know. And then we wanted to basically take these models further and train them on SARS-CoV-2 data, which is at which point you actually go into looking at the GenSlim model itself, which is trained on the SARS-CoV-2 data. And now we increase the sequence length to about 10,240, which is approximately 30,000 codons, which is great because this is the size of the SARS-CoV-2 genome. And then in order to actually place them in the contextual order, so think of it like a natural language description of what a book is all about. Within the book, you actually want to order the paragraphs in a certain order. And that's what this diffusion model allowed us to do, which is to build the same model, start with the foundation model that we had, and then go all the way to reordering the paragraphs. And this was something that we developed as part of the foundation model itself, because we could actually talk about relative ordering of the genes and where we want to put them and so on. The nice thing about this is this represents one of the largest foundation models that is trained on nucleotide sequences. I'm not going to talk a lot about the scaling and everything, which we'll talk during the tutorial section. But what is interesting from these models is that these models do reveal new biological insights. So I talked to you about the semantic embedding, and the semantic embeddings are the ones that are shown here. Every dot here is actually part of the validation data set. So this is not training data. And every dot here represents a gene that is from this validation data set that we are looking at. And now I'm going to paint that with certain quantities. Remember that this is a very high dimensional space that is now plunked down to this low dimensional representation. You can see the separation between low sequence length and high sequence length sequences. So that is nice because that's one of the things that you want the model to sort of understand. But more importantly, it can also capture the differences in terms of the GC content. So this clustering sort of highlights the fact that it can distinguish between low and high GC content, which means that it captures something about the fundamental adaptation of these genes to different environments. It's also interesting to note that these models capture something about the structure of these proteins or genes. And that's kind of cool because now we got the same structure, same protein, same function, biological function, but they have two different isoforms that are clearly separated in that landscape. Right? Given that we started completely from the raw nucleotides, the fact that it is able to capture these relationships, and not only that, it can also do this in the context of biological function. So many of these bacterial genes, you can see, they sort of cluster together as to how they have the different functions that are organized together. So in some sense, you can see the differences in the stress response and defense and virulence factors. You can also see several of the DNA processing elements together, membrane transport and things like that. So the fact that this model can sort of learn to distinguish different functions and also learn that from the raw nucleotide sequence was kind of cool, at least for us in terms of thinking through what we thought this model should be able to do. What is even more interesting is the fact that when you presented data completely from different forms of life. So for example, let's say I presented data from eukaryotes. This is not data that's part of the training at all, right? It has not seen this data at all. It can separate out coding versus non-coding sequences. It can separate out what constitutes mRNA, tRNA, and so on. And the fact that it is able to separate these out with such clarity, like reaching about 90 to 95% accuracy, depending on which model you choose, it's kind of nice to see that these models actually have strength in them in terms of understanding this. There is a comparative model where you can look at really large context windows, something that HYENA-DNA pioneered, which is one of the models from Stanford. And we can also see whether these models are able to look at the same set of questions across the different, this ones. And GenSlim is more or less comparative to, to these models. And the model that came out very recently called EVO, shout out to Brian High at Stanford, they compared a lot of the things that GenSlim baseline model was able to do with these, tasks, and we sort of perform closely aligned with what EVO also performs. EVO obviously is a much larger model compared to this model, which is 250 million or 2.5 billion parameters. So that's kind of cool. Now, the infrastructure for the foundation model, this is one of the instructors, Kyle, who is standing in front of all the three computers, it's fairly simple in the sense that we actually use Lightning, DeepSpeed, and Transformer libraries, right? And what is more interesting is the fact that you can actually download the model yourself, play with it. You can download the whole set of weights. Everything's available. The only thing that we request is that you download it via Globus, because that's much easier for us to manage and track and what's happening. And then you can basically do any of these sort of computations on top, playing with it. What is even more interesting is the fact that if you use a machine like Cerebrus, instead of having to train the model on Polaris supercomputer with 512 nodes or 2048 GPUs for approximately 2.5 weeks, we can end up getting much faster time to solution with just 4 CS2s, where we can train the model within about 10 hours or so, right? And you can also see the sort of scaling that we can do across the number of devices as we connect more and more CS2s, nearly 16x the performance in terms of scaling. And the model is also pretty good at predicting the various types of SARS-CoV-2 sequences. So it's kind of nice to see that these models actually can be used to do some more interesting stuff downstream. The next aspect is how do we do generative modeling with GenSlims? And this comes down to looking at reward modeling and reinforcement learning sort of ideas. And we said, can we use these together by incorporating experimental feedback, which is commonly one of the bigger challenges in biology, which is to say, how do I include experimental data into my pipeline? And so we have these pre-trained seed models that we talked about, the GenSlim model that I just spoke to you about. You can now define a set of intrinsic and extrinsic rewards. So the intrinsic rewards can be related to the sequence and the structure of the protein that you're interested in coding. And then you can have some aspects of extrinsic reward models, which can be either an assay that you develop inside the lab to measure, saying that this is actually something that I want to do via experimentation. And these models can then now be used to define this reward function that basically can be incorporated inside the RL loss function, or reinforcement learning sort of loss model. Now, the question becomes, does it work in practice? A very simple experiment for the model is to say, okay, generate me a malate dehydrogenase sequence that looks like malate dehydrogenase from one of the training data sets that I have. This is a very simple, zeroth order experiment that you want the model to succeed. And it gives you a protein that has 330 residues long. It has this very nice properties where it has all the conserved residues and so on. We can use OpenFold to kind of fold the structure. You can see the presence of the active site. It has conserved all these qualities, which is great. Now, I want to challenge it a little bit more by saying, okay, I'm going to give you a budget. Say I give you a budget of just 75% of the original sequence, and then ask you to design the same protein. It has to be a functional malate dehydrogenase. And so 75% of roughly 335 is 250 residues, give or take. And so you get a protein that is like 250 residues. You can see that you can fold the model using AlphaFold or OpenFold. You get the same site. You can run simulations to see if this is still functional in the computer to say, yes, it is functional. And now we are in the process of actually doing this experimental in the lab to kind of say, okay, this works. Can we actually make sure that this is assaying properly and so on? And one of the good things about it is that we had a 96 well plate that we ordered from a company. And out of the 96 well plates, out of the 96 constructs that we ordered, about 49 of them are functional malate dehydrogenases. This is a significantly higher yield than using many of the protein language models that you might use to actually design these proteins, right? And now if you use this in the context of like trying to say, what is the sort of challenge we face in computing terms? See, the generative models are kind of fun because they allow you to sort of look at parts of the landscape that you might have not really sampled. So the gray data is actually the training or natural sequences. Every colored dot that you see is the generated sequences from our code, from the model. But what I want you to observe is this last corner where there is a red dot on top, where we are exploring more novel sequences, it seems like. And those novel sequences are not part of the training data, or nor they're part of this generated sequence. They have some interesting properties about what they can do, which is the fact that they reduce the activation barrier by about 5 kcal per mole. Again, this is a very significant story because if it is true, if it works out in the lab and we can say that it is actually true, this basically tells you that we can explore new productive designs for the sort of rankings that you want to do on enzyme designs inside the lab. But this is coming at a huge computing cost, which is that even exploring like top 1%, say 1,000 variants, and when we are doing 20 simulation windows to actually simulate the reaction coordinates for this, the embedding space of the simulations can just overcome all of the computing needs of the supercomputers that we have, right? Hopefully on Aurora, we'll be able to simulate this on a single computer. Otherwise, we'll have to like basically distribute the loads across multiple supercomputing sites and do them. And labeling these productive designs and ranking is basically much larger compute requirement where the inference sort of handles its upper hand in terms of how we design them, right? Okay, so now how do we use these models in the context of moving on towards more interesting stuff on the genomic scale? How do we use this in the context of predicting new SARS-CoV-2 strains? And this is actually one of the things that we did in 2022, where we could show that this is possible, that we could train these foundation models, build a semantic score that basically emulates the sort of natural evolution, identify these sequences as having properties that mean that we need to pay attention to them as sort of a continuous watch list of these sequences. But what we challenged the model to do was to basically say, I'm going to present only 1 year's worth of data, meaning that we are going to show only 1 year's worth of the sequences. And that happens to be just alpha and beta strains from 2020. And then say, can you predict all the other strains for me? Right? That is a sort of a surrogate for saying it is able to show the evolutionary details of what is happening. And now what you're going to see in this movie is this clip where we have embedded every genome of SARS-CoV-2 that comes after 2021. And those things are now clustered nicely in this latent space. But we're going to paint them with something called as an alignment score, which is to say that, okay, it's doing something with respect to the sequence similarity correctly. But on top of it, we can actually now show the different strains, alpha, beta, delta, eta, gamma. And what is interesting to note here is that each of these strains are actually like nicely populated in their own clusters. But you also see the presence of multiple islands coming from the same strain that mostly corresponds to the countries from which they originated. And that was kind of cool because this metadata is actually not part of the original training data in any form. And now we can start to generate new sequences. Because we have the model, we can now start doing generation of these new sequences. What you see in gray are the natural sequences. What you see in red are the generated sequences. And they sort of follow the same phylogenetics of what the original strains looked like, which is good. And in some cases, they are actually like about 60 to 80 mutations away from the natural strains that were there, which again, from the model's perspective, is able to like show how the evolutionary trajectories might, propagate further. And then in 2022, when BQ1 was actually one of the strains that we were paying closer attention to, one of those strains indeed happened to be very close evolutionarily just by one mutation, which was away from BQ1, which kind of gave us more confidence that these models are actually more powerful than we actually thought or envisaged. So we can see this in the context of all the RBD, spike receptor binding domain, and the ACE2 interactions. It also shows you the mutations in yellow that kind of highlights how they are related to each other and why we should be paying attention to them. And again, without touching the model, we said, without even fine-tuning the models further downstream over the course of the years that we have seen, we asked, is it able to see EG5 and XBB 1.5? Again, depending on the parameters that you set for the generative model, you can actually bound how closely or tightly it is to the wild-type sequences. And what you see in the different colors are the predictions that are made by the model in terms of where it is varying. And what I want to highlight is that the grays are the ones that match exactly with the wild-type sequence. The oranges basically represent substitutions, should be done. And the blue ones are the ones that we get wrongly in terms of our corrections. The last one, I'm going to basically jump over a lot of details, but it's basically to use these generative models as embodied agents where you can actionize these models to have different tools. This is part of an initiative at Argonne, what we call as autonomous discovery. And the story begins with this nice tool set where we can actually have digital twins of all the things that we want to do inside the lab. And if you've done molecular biology, you can actually think of this as a PCR, polymerase chain reaction model that's being run both simultaneously in the twin model, digital twin model, as well as the robotic infrastructure that we have. And this is actually to have the AI and robotics and simulations in a closed loop so that we can plan these experiments and run them and so on. Again, there's a lot of work that needs to happen in order to get to this point where we can plan and run experiments. Again, this is used in the context of designing antimicrobial peptides, but we have come to the point where we can actually generate new protocols inside the lab. So this is an agent for trying to say how we can do automated lab generation, automated lab code generation process. This is led by Brian Su in the lab, where we have an agent that sort of looks at the task prompt of saying, I want to be able to do PCR in the lab. And then it basically breaks it down into this hierarchy of tasks. And those tasks get into this model where we can actually start looking at candidate code and refining them further and further. And as it finds more and more code that actually works with the model, it can add them as skills and recall those skills whenever it wants to start running those skills and retrieve them on the basis of what it needs to run. So this iterative model of actually generating code and having it run in a way that can execute an entire biological protocol is actually one of the stronger basics. And you'll hear a lot from Ian talking about this infrastructure and how he's envisaging this whole thing, connecting the different biological labs and material science labs and all those things together. But we are also very much into training the next generation of people that want to use this sort of autonomous labs. So anybody that is interested in it, please do come and talk to me. We have summer programs, we have lectures, we have virtual reality environments where you can soak yourself in and try to run these experiments yourself. And we want to make sure that it is targeted at everybody that can use this infrastructure. So in summary, these models that we are trying to build have a lot of implications for health and biomedicine. They're good for integrating experimental observations in a different manner than we are used to thinking about it in general. Of course, there are several challenges that exist, including the fact that we need to align these models with respect to knowledge that we know about genes and functions and proteins. We need to scale these models to have much longer context windows, especially when you're thinking about even bacterial genomes or even eukaryotes. You want to integrate this with text and other modalities, including imaging and high throughput experiments, where we are starting to take more baby steps. And then finally, I want to thank everybody, including my collaborators, both at Caltech. Anima is not at NVIDIA anymore, but she's still a very close collaborator of ours in the biospace. Cerebrus people who scaled all of this on their machines and showed that it's possible to do it. My entire group and people that are involved and collaborators across the lab, and the Viz and the data people who have been really instrumental, and the autonomous lab group that has been standing up these robotic instruments. So with that, thank you so much for your attention. And
what a beautiful talk that combines biology, computer science, AI, HPC, and robotics. It's very impressive. And I do want to say that Arvind's group is quite big, and they work on a variety of things, and we are starting a collaboration with them in a very big way this summer. And there's going to be at least 10 interns here. You can switch. From Michigan that are going to be working with this group. So we have time for one question, but then as Arvind said, they're going to give the tutorial, and Ian's talk tomorrow at 1:30 p.m. will paint a broader picture of where they're going. So one question, please go ahead.
I'm Gil Lohman in the medical school. Very nice talk, of course. You specifically ended on antimicrobial agents, which is a huge international problem.
Yep.
I would like to know how you organize the approach to detect not just successful targeting of a known target, but the avoidance of adverse effects, which could be much broader range of unknown targets as detected in animals and humans.
Great question. So there's a lot of external data integration that has to happen within these models. Currently, the goal of this lab, as I would call it, is to basically generate exemplars that show promise inside the bacterial samples, meaning that we get some results in terms of their ability to inhibit the growth of that particular bacteria. And the reason for choosing this sort of a style was that we know downstream how to handle all the other tasks of saying cross-adversarial effects or other sort of effects that we need to be worried about. And we are using the AI to kind of already filter a lot of the results in terms of new hypothesis, which is coming out of these models that tell us whether we should be paying attention to any sort of cross interactions and so on. This is what the AI is really good at doing, really speaking. It can sort of identify across a plethora of species whether there are cross interactions that we need to be worried about. And the failures actually constitutes preferences for these models to basically rationalize the generation process. So if you want to do the generation by paying attention to the failures, we can actually reduce the number of these sort of structures that we generate from these models. So there is 2 levels of tuning that's happening, which is kind of interesting from the perspective of how we think the AI should work. So happy to take that even more in detail and explain as to how we are thinking through that. And would love to have people's views on it, because I know there's a lot of people working on veterinary sciences where there's an interest in like doing this at scale. So yeah.
Very good. I think there are many more different kinds of systems across all the different organ targets, off target, than for just the microbial actions.
Correct. Thanks.
Thank you very much.